Metformin was never intended to extend life. Synthesized in the 1920s and introduced in the 1950s as a treatment for type 2 diabetes, it has since become one of the most prescribed drugs in the world. Its purpose was simple: to lower blood glucose. Yet over the decades, researchers noticed something unexpected. This old and inexpensive molecule seemed to influence how the body ages. The first clue came from clinical evidence rather than theory. In 1977, the United Kingdom prospective diabetes study began, a landmark randomized controlled trial that followed more than 5,000 newly diagnosed patients with type 2 diabetes for up to 20 years. Among overweight participants, those assigned to metformin monotherapy had a 36% lower risk of all-cause mortality, 42% lower diabetes-related mortality, and 39% lower risk of myocardial infarction compared with conventional treatment. Even after the trial ended, the survival advantage persisted for at least a decade, a phenomenon researchers called the legacy effect. It was the first controlled human evidence suggesting that metabolic intervention could alter not just disease progression, but the trajectory of lifespan itself. However, later trials painted a more cautious picture. The Diabetes Prevention Program, launched in 1996, enrolled 3,234 adults with impaired glucose tolerance, people at high risk but not yet diabetic. Participants were randomly assigned to receive either lifestyle modification, metformin, or placebo. After 3 years, metformin reduced the incidence of diabetes by 31% compared with placebo, but mortality was not significantly different among groups. Its long-term follow-up, the DPP outcome study, extended the observation to over 20 years and confirmed the same pattern. Metformin delayed the onset of diabetes but did not reduce all-cause, cardiovascular, or cancer mortality. When data from multiple randomized controlled trials were pooled in meta-analyses, the overall conclusion was consistent. Metformin does not significantly extend lifespan in humans, at least within the context of existing trials. Outside of randomized studies, observational data told a more ambiguous story. In 2014, Bannister and colleagues analysed health records from the UK Clinical Practice Research Data Link, including more than 180,000 adults with type 2 diabetes. They compared patients on metformin monotherapy with those on sulfonylurea and also with non-diabetic controls matched for age and sex. The results were striking. Metformin users had a median survival 15% longer than non-diabetic controls, while sulfonylurea users had shorter survival. After adjustment for confounding factors, metformin use remained associated with a 15–20% lower risk of death. However, researchers cautioned that these findings might reflect selection bias and immortal time bias. Healthy patients are more likely to be prescribed metformin, and longer survival can be a statistical artifact rather than a biological effect. When such biases were corrected in later analyses, the survival advantage often weakened or disappeared. Despite these uncertainties, metformin's biological effects remain of great interest. At the cellular level, it activates AMP-activated protein kinase, inhibits mTOR signaling, reduces oxidative stress, and improves mitochondrial efficiency. These pathways are central to the biology of aging, and in yeast, nematodes, and rodents, metformin has been shown to extend lifespan and delay age-related decline. In humans, however, translating these molecular benefits into measurable longevity has proven difficult. Aging is not a single process to be switched off. It is the accumulation of molecular failures across time. To address these questions, scientists designed the Targeting Aging with Metformin trial, the first large-scale human study to test whether a drug can delay aging itself. TAME aims to recruit around 3,000 adults aged 65-79 across the United States and follow them for 6 years. Rather than measuring lifespan directly, the trial's primary endpoint is the time to occurrence of any major age-related disease, including cardiovascular disease, cancer, cognitive decline, or physical frailty. By evaluating whether metformin can postpone the onset of these conditions simultaneously, TAME seeks to determine if aging can be treated as a unified biological process rather than a series of separate diseases. The trial is ongoing, and its results may reshape how medicine defines and measures aging. The paradox remains. Metformin modifies many of the biochemical hallmarks of aging, yet has not consistently extended human life. Its effects seem to slow certain biological processes, but not the final outcome. This distinction highlights a deeper truth. Slowing disease is not the same as slowing time. The drug's greatest value may lie not in conquering mortality, but in compressing morbidity, extending the healthy span of life, not its total length. In that sense, metformin is not a key to immortality, but a calibration of decline. It offers a reminder that the purpose of longevity research is not to stop the clock, but to learn how to use the time we already have more wisely.